Hey guys, it's Josh. Welcome back to another MCAT question of the day. Let's hop right into it.
This question says, which peptide sequence is most likely found in a transmembrane helix of a protein? And we're given a bunch of different strings of amino acids. So before we analyze the answer choices, let's try to visualize what's going on and come up with a prediction for what we expect to see. So if we're focused on a peptide sequence that's in this transmembrane helix of a protein, we're probably looking for a nonpolar string of amino acids, right? So if we have this plasma membrane and we got our polar heads and we got these nonpolar tails, we know this transmembrane domain, this right here where that protein is going to be located, that is a nonpolar hydrophobic region. And we know that the extracellular matrix is usually a polar region and the cytosol is also a polar region. And we know this protein is a transmembrane protein. So that's going to be stuck right in here. So in that nonpolar region. So we're going to want to look for an answer choice that has nonpolar amino acids involved. So if I was taking the MCAT test, what I would do is I just look at the amino acids and figure out if there's a charged one, let's rule it off. So option choice A, we got alanine, isoleucine, phenylalanine, valine, and leucine. All of those are nonpolar amino acids, so that one is looking pretty good. I'm going to keep that in the running for now. Alanine, option choice B. We got alanine, threonine, lysine, asparagine, leucine. So lysine is one of our positively charged amino acids, and threonine we know is polar. And asparagine is polar as well. Because of that, this is not a very good answer choice because we're going to have no charges in that transmembrane domain. So let's rule off option B. Option choice C, we got lysine, threonine, arginine, asparagine, and histidine. So this option is even worse. Lysine is positively charged, histidine is positively charged, threonine is polar, arginine is positively charged, and asparagine is also polar. So that is even worse than option choice B. Let's rule that out. Option choice D, we got valine, threonine, proline, tyrosine, and serine. So valine is nonpolar, that's good. Threonine is polar, proline is nonpolar, tyrosine is polar, and serine is polar. So we don't have any charges, but that's definitely not as good as option choice A, where we have all nonpolar amino acids. So I'm going to rule off option choice D, and let's go with option choice A in this scenario.